name's Lachlan. I'm from the Doherty Institute at the University of Melbourne, and I'm going to present my poster on a strategy for whole genome sequencing of tuberculosis DNA directly from sputum without culture. Mycobacterium tuberculosis is a very slow-growing bacteria, meaning that to get enough DNA to be able to perform whole genome sequencing, the bacteria needs to be cultured for at least 2 weeks and typically longer. And that's a problem because of the high rates of drug resistance in Mycobacterium tuberculosis. It's necessary to grow the bacteria in order to be able to predict the drug resistance. And that's necessary in order to make sure that the patients are getting the correct treatment early on. And given the long time it takes in order to get that amount of DNA, that delays use of appropriate treatment. So while there are methods in place for PCR-based methods for detecting specific resistance mutations, these don't cover the entire genome. They miss some resistance mutations outside of the target regions, and they would miss resistant mutations in novel or recently described resistance genes. So our approach is to try to capture the Mycobacterium tuberculosis, the whole bacteria, directly from the sputum, followed by DNA extraction of the captured mycobacteria and sequencing of that DNA. So in order to do that, we have designed magnetic nanoparticles which are functionalized with antibodies which recognize the mycolic acid present in the cell wall of mycobacteria. So we use those magnetic nanoparticles, we put them in the sputum which has already been treated with NaOH to remove other contaminants, and then we use a magnetic block to basically capture those magnetic particles and enrich the mycobacterium. So using spike-in experiments with an avirulent strain of MTB, where we spike in this avirulent MTB into healthy sputum from a non-tuberculosis infected patient, we can control the amount of sputum which is present. So we spike in a different range of concentrations, but up to 10 to the 5 CFU per mil. And we show that with increasing concentration of these magnetic nanoparticles, we can capture up to 70% of the mycobacterium which we've spiked into that sputum. The next step, so because we start with a relatively small amount of mycobacteria, even if we capture 70%, we still get sub-nanogram levels of DNA out of that. So in order to have enough DNA for sequencing, we need to amplify that DNA. So we use a whole genome isothermal amplification approach from QIAGEN called Genomify, which uses the Phi-29 DNA polymerase in combination with random primers, which would bind randomly across the genome to attempt to uniformly amplify the small amount of DNA. You can see here that we start with sub-nanogram levels of avirulent MTB, and after amplification, we end up with thousands of nanograms. So that's a highly effective method for amplifying. It's also quite, has a high fidelity. So even if we start with a small number of copies of MTB DNA, we're quite confident that it's not going to introduce too many mutations and not going to skew our results at the end. So once we've extracted and amplified the DNA, we use an Oxford nanopore ligation kit to ligate the adapters for nanopore sequencing, and then we can put it on a gridiron, for example, or a minion to sequence the DNA. And then we have also designed a customized streaming bioinformatics platform which carries out the mapping and identification of variants from the stream of data that comes off the nanopore sequencer. So we like nanopore sequencing both because it's a very portable sequencing approach, but also because you get data off the machine almost immediately as you start sequencing in a data stream, and we process that data stream in order to identify the presence of antibiotic resistance mutations in our data, and also to predict the lineage and the strain. And you see from this, we can identify the proportion of reads which we get after this process, which map to the MTB genome. And so this shows that when we use the nanoparticles in combination with our amplification approach, we get almost entirely mycobacterial DNA out of it. We didn't get anything if we didn't use the nanoparticles. We didn't get sufficient initial concentration. And this just shows that with the magnetic particles at a admittedly quite high spike in 10 to the 5, we get similar results to culture with or without the nanoparticles. So we can use culture with nanoparticles or without, it doesn't matter because we have a high starting material to begin with. And then this just shows when we map the reads to the genome, we get, it's not completely uniform coverage because this 529 can kind of amplify things a little unevenly, but we still get decent coverage of above 30x for all the resistance genes we're interested in. Some genes have a much higher coverage of over 200x, but we get a decent coverage of the genome, and we can from that figure out what the resistance mutations are in the sample. So just to summarize, we call this method a duo enrichment strategy because we've got the capture followed by the whole genome amplification, and it's a promising method for culture-free sequencing of mycobacterial tuberculosis. I'd like to specifically acknowledge Dr. Anne Yuen, who's carried out most of the experiments in this work, as well as Sanjay KC, who designed the magnetic nanoparticles. Michael Hall developed a bioinformatics pipeline. Jianshu Zhang helped run that bioinformatics pipeline. Arnold Banamugiza has provided us with material and also helped design the antibodies for the nanoparticles. And Mark Blaskovich has also been involved in the design of the nanoparticles. And this work has been funded by the Medical Research Foundation of Australia. Thank you.